we go. So I needed to unmute myself, which is probably useful for a talk. Hopefully I'm audible now though. So yeah, this is some work I've done with Jennifer Hoyle-Cuthill. And probably I'm at the beginning of this session because parsimony is the most primitive form of phylogenetics, but maybe I will convince you that there are still some interesting questions out there. So as a sort of general motivation for this, so my co-author has previously done some work looking at maximum possible steps for a multi-state unordered character, and even managed to prove that what she'd done as a formula was that, like, mathematically prove that it was the maximum case. So as someone that spent far too much time looking at morphological codistic data sets, I wondered, can we generalize this to the broader forms of characters that actually exist out there? So an example here is on the right. Oh, something's popped up for me. Just gonna hit return. Okay, hopefully that's fine. So we're going to do generalized maximum parsimony. We have to use the cost matrix to do that. So most of you probably know what a cost matrix is, but briefly, it's a square matrix where rows and columns are the same. So they have the same state labels. And then the individual cells are whatever the cost is to go from a state, which is by convention the row, to another state, which is by convention the column. So how you do this in maximum parsimony was first derived by Sankoff, but there's a much nicer description, or at least for comprehensibility for people like me, in this book chapter by Swafford and Madison. So I recommend reading that if you're interested. So before I sort of get into more details, what probably useful at this stage to point out is that there's a link between the cost matrices and graphs, so graph theory. So here we have an example cost matrix in the upper left. So we could represent that as we do in the upper right here with a directed graph where we've got basically every single off diagonal value is an arc on a directed graph. There's a lot of redundancy in this. So we can see all those costs are symmetric, for example. So we could instead do in the bottom right a graph representation, i.e. undirected, because of all those symmetries, but also that transition between 0 and 2, that information of it costing 2 is kind of already covered by the indirect transition via state 1. So on the bottom left is a minimum graph representation that basically covers all the information from the cost matrix. So I'm just going to state here we're explicitly assuming the graph representation, the minimum graph representation of the cost matrix is the graph representation. So there's sort of a bunch of background before I get more into the actual limits and proofs, but I'm going to start by talking about 7 rules that cost matrix must meet to be valid, show a classification of cladistic characters into 12 different types, and then talk about 3 different limits that we're interested in. So for each rule, they're numbered 1 to 7. I'm going to show a valid example on the left and an invalid example on the right. So the first is simply that all costs must be specified. So what that really means is all the off-diagonal values are a degree of freedom. You can't know what they are, even if you know all the other costs. The second rule, this turns out to be quite important for a lot of the maths, is that the diagonal must always be 0. So you can't ever be a cost for staying in the same state, effectively a non-transition. By contrast, every off-diagonal does have to have a positive transition cost, because you are actually moving from one state to another. But these could, in some cases, be infinite. So another important thing is when we consider the state graph, it has to be connected. And that's because ultimately we need to be able to map to a phylogenetic tree, which is by definition a connected graph. So we can't have examples like on the right here. If we want to force a particular root state, we can do that by setting cost matrix columns to all infinity values except for the diagonal. But of course, given we again want to map to a phylogenetic tree, we can only do that for one state. So 2 in the valid example on the left here. And then it's a bit subtly different, but another one is at least one state must be permitted to be the root. What this means in graph theory terms is you have to have single source, multi-target, a graph, and that effectively means there must be at least one state that can be the root, and it must be able to reach all the other states. And then one that's been recognized for a long time is the costs must be self-consistent. So they must basically meet triangle equality. So this is really well known. So it goes back to Wheeler 1993. And if you use software like Help or TNT, it will automatically check for and correct your cost matrix if you violate this one. So these are just mathematical rules. There's no empirical things that I know of that break them, but they're useful for reference for thinking about the questions in here. So we like classifications in biology, so I'm going to classify cladistic characters into 12 types. They're going to have a Roman numeral and a name. Then I'm going to use an empirical example in the middle here. So show the cost matrix on the left, the state graph on the right, and if you really want to know the anatomy, the character number and reference below that. And then for interest, the empirical proportions of those at the bottom. So first of all, just for completeness, this is a constant character, it's pretty boring, only one state, but it is actually, strangely enough, in morphological data sets quite common. Next is binary symmetric cost. So typically this will be 1, but it doesn't have to be to meet the criterion, just has to be symmetric and finite, basically. Then there's multi-state unordered symmetric. So this is basically where every state can directly access every other state. So we have the complete graph as our state graph. Then we get linear ordered, where we basically have all our states are in a row. So there's intermediate states in between, and all the costs again are symmetric, and the weights of our edges have to be equal. So we can also have non-linear ordered graphs. So this means you have at least one branching point. So you have to have at least 4 states for that to be possible. But again, all the connections basically are equal weight. Then we can consider asymmetric examples here. So here's an irreversible. So typically here, the bottom left of our cost matrix will all be infinities. It could be that the upper right, though, would still meet the criteria. So basically, all transitions have to be in a particular direction. So you're a priori setting the root state effectively. And then we can extend that to multi-state. The important sort of constraint here is that they have to be linear in effect. If you want to branch, you'd need to define a more complex character. So I'm going to skip over the next 2 because these are related to Dollo, which turns out to be a special case rather than a general case. And then type X is where we have the most complex form of symmetric characters, where essentially it's like an ordered, where you can have branching, you can have cycles, but there has to be at least, when we draw the state graph, 2 different edge weights out there, so they're not all the same. Then we can have binary custom, that should say asymmetric. Where you've got finite costs, but they're different going forwards and backwards. And then probably the most general in scope would be type 12. So this is multi-state custom asymmetric. So at least one set of costs are asymmetric, but they don't all have to be here. So 0 and 1 are symmetric, but between 0 and 2 and 1 and 2 are asymmetric. So the 3 limits we're interested in is for these, applying to these is the minimum cost. So here we only need to consider a cost matrix, because the state frequencies don't matter, because we're just going to group them conceptually together, all the zeros together, all the ones together, etc. We want to maximize our costs. We're considering G, so we've got a restriction of a cost matrix and specific state frequencies. And then if we want to maximize G, we're sticking with the cost matrix, but we're considering what state frequency would maximize that cost. So before we start, this is effectively the state before we did any research. There's already a bunch of proofs out there that cover at least part of these, so the 3 limits and our 12 character types. So what we tried to do basically is turn those remaining red crosses into green ticks. So for the minimum cost, our argument here is that this is already a solved problem in graph theory. It's simply the minimum spanning tree length. So we take any valid cost matrix, so meeting our 7 rules, we find the minimum spanning tree or a minimum spanning tree, and then we simply take the length of the tree. So this is already a sort of well understood mathematical thing. So it's basically already out there, and we just need to make the connection between them. So this nicely covers basically the remaining gaps in M. Again, I'm leaving Dolo out here because that turns out to be a special case. So for G, which is our maximum cost, again, this is already solved as a general case, and it's solved by looking at the star tree. So star trees are nice because they're really simple to deal with. We already have a single internal node. We just have to find whatever state gives us the minimum cost because we're doing maximum parsimony, and that is going to set the overall maximum cost that we can realize. So this has already been proved. So there's a proof, for example, in Golubov's book from last year, and I'm briefly going to show you how that proof works. So formally to state it, what we care about is that there's no longer most parsimonious tree than the star tree. We don't care if there are other trees the same length. So if we want to prove that, we can start with any valid cost matrix. I'm using a binary example here. Then we have any set of states. And then we start by working out what would be the most parsimonious root state. If there are multiple most parsimonious roots, you can pick any of them, it doesn't matter. Then we consider the case of any other non-star tree. So it doesn't have to be fully bifurcating, or in this example it is. Then we can set all our internal nodes as the most parsimonious root state on our star tree. So if we ask the question, what's the length of this tree using our cost matrix? Hopefully it should be clear this would be the same as the star tree. And that's because if we look at any internal branch, it's going from the same state to itself, basically. So under rule 2, that always has to have cost 0. So our internal branches will always add up to 0. Then if we look at our terminals, where the cost we calculate must be, oops, I am way over time. We find that that they're always going to go from the root state to the tip state, basically. So this will mean we have the same length as our star tree. So if we then consider any other set of internal nodes, we will see that there are only 3 possible outcomes when we calculate the length of this tree. So it can be shorter or the same, which we don't worry about because that can't be longer. But we also don't need to worry about the case where it's greater, because that cannot be a most parsimonious solution, because we have already shown that there is a more parsimonious solution by setting every internal node to be the same as our star tree. So that basically solves the rest of them, but now I'm probably going to have to skip quite a bit because I'm way over time. So yeah, so GMAX, we have a couple of different solutions here. So one is just to exhaustively look at things, and that works for a lot of empirical examples, but it's obviously not very nice mathematically. So a nicer way to do it algebraically is to find, if we set one of our 2 states as the root, we simply ask what state frequency gets maximized. So that's always going to be at the edges, but it's the point where these things cross, which is going to be the most parsimonious solution that sets basically our maximum cost. I'm probably just going to have to skip to the end though. So there's a lot of other stuff in here. But yeah, so if I'd have got through it in more time, I did show you 7 rules for mathematically define a valid cost matrix. I classified them into 12 different types. And then we reviewed or provided proofs, although some of them were skipped, for those 3 limits.
All right, Graham, I'm going to have to stop you there. I'm sorry.
Yeah, sorry.
No problem.
Okay.
Graham will be here for the rest of the session. So if you do have questions, you can approach him in the chat, or if you want to ask them publicly, you can approach those and put them in the chat, or you can directly message him. All right, wonderful. And our next speaker is ready, and we can see her slides perfectly. All right, take it away.
Hi everyone, I'm Zhao Xingwu, and I recently graduated from the University of Wisconsin-Madison. I did my undergraduate there, and today I want to share with you a method that we developed to infer phylogenetic network using algebraic invariance. As many of you might know that the tree of life is a metaphor and model that is used to explore the evolution of life. It can be represented by a phylogenetic tree, where each node represents each organism and each edge indicates the relationships between organisms. But this type of structure is unable to capture some important biological realities like hybridization, integration, or horizontal gene transfer. So nowadays, people prefer to use phylogenetic network to represent the tree of life because it is able to account for the reticulate evolution. And here's an example of the network representing dog. The network method is actually an extension to the tree structure by the explicit modeling of gene flow. So there is also an unrooted version of the phylogenetic network where the position of the root node is suppressed and the direction of the edges is suppressed. Because the placement of the root is constrained, the direction of the hybrid edges actually can inform some information about the direction of time at this node. There are several existing methods to infer phylogenetic network. PhyloNet is the pioneer of likelihood inference of phylogenetic network, but it is unable to handle dozens of taxa and hundreds of genes. SNAC is inside the PhyloNetwork Julia package, and it is a pseudo-likelihood model under the coalescent model. The running time does not increase as the number of genes increase, but it is still not scalable as the number of taxa increase. This too is a co-estimation method that infers simultaneously the phylogenetic network and the gene trees, but the complexity still makes it unsuitable to handle big data set. So due to the limits of all existing methods, our goal here is to develop an inference method for phylogenetic network that is scalable for large data set by using something we call algebraic invariance. So our work focus on this type of structure that we call 4-node hybridization cycle on a level 1 phylogenetic network. This structure we refer to it as a diamond. So here is a unrooted representation of this type of network where the hybrid node and hybrid edges are denoted in blue. T here represent the branch length, and each node N0, N1, N2, and N3 represent a clade in the network. Here are some specific examples of this diamond structure. For example, for the diamond structure for A taxa, we have 2 nodes in each 2 taxa in each clade, and we refer this type of structure as N2222. Here's another example. For 6 taxa, here we have 2 taxa in N0, 2 taxa in N1, 1 in N2, and 1 in N3. So we denote this as N2211. This notation is going to be helpful for my further explanation. So to infer the network with diamond structure, we already know that the distribution of the gene trees provides some information on the true network that generates the data. Our previous work has also shown that the split frequency of subsets of 4 taxa also provide information on the true network. And we name this split frequency of subsets of 4 taxa as concordance factor. Our proposed method is going to take the concordance factor as input. Before I move on to explain what is concordance factor, I want to emphasize that our method is also able to take in other types of input. For example, if the biologists have the DNA sequence by their hand, our method can use IQ tree to infer gene trees from the DNA sequence. And then we can also use Philo network as a Julia package to extract concordance factors from the gene trees. So in total, our method is able to take in the gene sequence, gene trees, and also concordance factors as input. So what is concordance factor? For each subset of 4 taxa, for instance, A, B, C, D, there are 3 possible splits, A, B, and C, D, A, C, and B, D, A, D, and B, C. We call these splits quartets. A concordance factor is just the proportion of gene trees that display this type of structure. And we can represent each concordance factor as a polynomial equation as shown here with respect to their branch lengths. So our method is going to take in algebraic invariants, which are generated from those concordance factor polynomials. To get these invariants, we can sample all possible 4 taxon subsets and find a set of polynomial equation that represents the proportion of gene trees displaying all possible quartets, so that we can get a set of concordance factor formulas, CF of n. Next, we associate each concordance factor polynomial with a variable AI. Here, we want to identify the relationships that these AI values need to satisfy for the polynomial equation system to be consistent. And these relationships, we'll denote this as algebraic invariants. This might be a bit overwhelming to hear, but let me give you an example. So the concordance factors from a given 4 taxon subset need to sum up to 1. So here we have A1 plus A2 plus A3 is equal to 1. This is a relationship we already know. So here the invariant is very simple. It's simply A1 plus A2 plus A3 minus 1 is equal to 0. This is just a very simple example, but for some other cases, the invariant equation can be very complicated. And sometimes it can be very daunting like this, but luckily we have software to help us calculate these equations, and we do not need to do it by hand. So we can generate the set of invariants for all possible candidate network. And we just need to evaluate our data with all these invariants to determine the candidate network that best matches with our data. So now here's the question. What is the candidate network? Does that mean it's the entire network space? But luckily, previous work have shown that we only need 2 taxa per clade to represent all concordance factor polynomials involving the hybridization cycle. That means we do not need to use the entire network space as our candidate network. We can just use this network with networks less than or equal to 8 taxa. In this way, we can get the set of polynomial invariants for each network. So let me explain the entire procedure of our inference method. We divide it into 2 different cases. First, for the case of fewer than or equal to 8 taxa, we evaluate our data on the invariants for all candidate networks. And the candidate network with the smallest invariant score is the network that best matches with our data. The second case is more than 8 taxa. Our method is going to iterate through all possible subsets of 8 taxa and then fit our phylogenetic invariants on each subset to identify the top subnetwork with 8 taxa, and then add the taxa individually, the remaining taxa individually to the top subnetwork according to the invariance score. So our method doesn't need to search the entire network space, and also it only needs to evaluate the algebraic invariant equations. So it is optimization free, and naturally in theory it should be faster than other methods. So we test our method in different simulation scenarios, and the results are all pretty good. We tested with true concurrence factor table and also with concurrence factor table perturbed by different levels of Gaussian noise. Our method also performs well with simulated gene trees and also with simulated DNA sequence. Here is a comparison of the running time of our method with other methods. So this is tested on the data of eight species. And we find out that our method is more than 10 times faster than some of the other methods. We also test our method on a real data set, the Canis genus. And the runtime is still faster than other methods. And in terms of the output network, our method is able to identify the correct network as in the Canis genus. But for example, in the Phylogenetics method, it is not able to identify the correct network. So in conclusion, our method use algebraic invariance, and in this way, it is able to avoid optimization and avoid searching the large network space. It is faster than a lot of the other methods. And in the future work, we hope to test our method with more species. And our work is now available in arXiv, and our method is available in the Julia package Phylodiamond. I want to thank my lab members for their help, and thank you all for your listening.
Wonderful. If anyone has questions for Dr. Wu, you are welcome to either put them in the chat if you're feeling shy, or you can put your hand up and I will authorize you to unmute and you can ask your question. All right, well, it looks like we are ready to move on to Dr. Bonsal's talk. After this, we do have a withdrawn talk, so we will have a short break following. All right, take it away, Dr. Bonsal.
Great, thank you very much. So my name is Mukul Bansal, and this talk is about some recently published research paper. It's about improving phylogenetic dating or better reconstruction of chronograms. So before I begin, let me just acknowledge the lead author and the collaborators and the funding agency. So Abhijit Mandal is the person who actually did most of this work. He graduated a few months ago, so he's not here anymore. And our collaborators at MIT, Greg, Tiberio, and Jack, and of course, NSF for funding this work. So the overall goal of this work is to construct more accurate chronograms. And I assume we all know what a chronogram means. So a chronogram is basically a dated phylogeny, right? Or also sometimes referred to as a time tree. So a chronogram is just a dated phylogenetic tree whose branch lengths have been scaled to represent time. And so here's a simple example of a chronogram. And here, so you can see that, so here we assume that the leaves are all present day species. They're the present time. And the branch lengths given in blue here represent time. So 1 means 1 million years, 4 means 4 million years, and so on. And so a chronogram basically labels the each branch of a phylogenetic tree like this with time. And based on that, you can also then infer that this node Z existed 1 million years ago and Y existed 2 million years ago, and so on. So this is what we want to reconstruct, and we want to do this sort of more accurately. Now, to reconstruct a chronogram, we typically need 3 things. So first, we need a regular phylogenetic tree, like a maximum likelihood tree or something. And in this kind of a tree, the branch lengths are expressed in terms of substitutions per site, right? The average number of substitutions per site. And so that's one of the inputs here. In addition, we need some sort of date estimate, right, for at least one of the nodes of the phylogeny, preferably more than just one node of the phylogeny. And this is what you can then use to scale the branches of the given phylogenetic tree to actual time units. And the third thing you need is an appropriate molecular clock model, right? So rates of evolution along the tree can vary both with time and with lineages. And so molecular clock models account for that sort of rate variation. And so you need an appropriate clock model to do this kind of inference. So here's a simple example, right? So for example, if suppose this is your input phylogenetic tree in which the branch lengths are in terms of substitutions per site. And then suppose you're also given information about date estimates for some of the nodes. So we know that the leaves represent present day species, that node Y existed between 6 million and 9 million years ago, and that X is at least 12 million years old. Then the output of chronogram inference would be this sort of a chronogram that scales the branches of this tree to represent time, right? So this, for example, this branch ZA is now 3 million years, represents 3 million years. And it satisfies all of these constraints, the date constraints. Now, in the past couple of decades or so, there have been many advances in chronogram inference methodology. So there have been great improvements in molecular clock models. There have been better ways to incorporate node age calibration. So for example, you can specify distributions of dates for each node rather than just a hard upper bound, lower bound, and things like that. And there's also improvements to just the algorithmics of inference itself. However, one thing that's still lacking is that these traditional methods for chronogram inference cannot yet use relative time constraints. So a relative time constraint is one that says that sort of gives you a relationship between two nodes of that phylogeny. So it's not telling you this node lived between 10 million and 15 million years ago. It's just saying, oh, this node must be at least as old as some other node in that tree, right? So a node X must be at least as old as some node Y. And these kinds of constraints can often be confidently inferred based on past horizontal gene transfer events, for example. But there is no good way right now to incorporate this information to improve chronogram reconstruction. So here's a simple example, right? So suppose I have this species tree, and I know that there's been a horizontal gene transfer event from this edge ZA to this edge XV, then this implies that node Z must be at least as old as node V. If not, then this transfer would actually imply sort of a backward in time flow of information, right? Which is impossible. So So horizontal gene transfers give us some information about coexistence of species. So that's what we want to do, right? So we want to use relative time constraints to improve chronogram reconstruction. And it is actually well known that chronograms inferred using only traditional node age calibrations often violate these relative time constraints. So, for example, if you apply some method to a phylogenetic tree, then the inferred chronogram might violate this known relative constraint, which says that node Y must be at least as old as node W. And so the goal is that we want to be able to combine both types of calibrations, node age calibrations as well as relative time constraints, to do better dating, right? So perhaps if you combine both, then you can infer a more accurate dating that satisfies everything, satisfies the node age calibrations as well as the relative dating. And this, of course, is particularly important for microbes because there is not much fossil data in microbes. So dating microbial phylogenies is very, very difficult. And on the flip side, horizontal gene transfer is plentiful in microbes, right? So you can actually use that information to greatly improve chronograms for microbial species. Now, there's been some work on this problem in recent years. So starting in 2018, there were these very simple approaches that attempted to use horizontal gene transfers to transfer node age calibrations, right? So that's a very simple way to do this. So you say, oh, if X is older than Y, and I know that Y is at least 12 million years old, then I can maybe say that X is also at least 12 million years old, right? So the first few papers in this series actually were just doing those kinds of very simple things. And later, some more complicated approaches were developed as well. Now, the approach most closely related to ours, to Dater, is the Bayesian rejection sampling approach from Fournier et al. 2021. And what this approach basically does, it's very simple. So it basically runs an existing Bayesian inference approach to infer chronograms, and then so it samples a bunch of chronograms from the posterior under some appropriate model, and then and this is done using only the node age calibrations, right? So not the relative calibration. So use some existing method to sample these chronograms, and then you just reject the chronograms that do not satisfy all or most of your relative constraints. And so once you do this, you're left with some small collection of samples, and then you just aggregate over those unrejected chronograms. Now, this approach has many obvious limitations. In fact, often what we find is that there aren't any samples that satisfy all the relative constraints. So pretty much all the time, you have to relax the requirement that all relative constraints have to be satisfied. And so even in the final phylogeny, the final chronogram, you will have violated constraints. And just to generate enough samples that are unrejected, you have to extend the sampling time. So these approaches often have to be run for very, very long just to get enough samples. So Dater builds on this kind of simple idea, and it also uses Bayesian posterior samples, but the idea is to error correct each sample instead of doing a rejection or acceptance, right? So even if the sample does not satisfy all constraints, relative constraints, you can error correct it and use it rather than rejecting it. So the key idea behind Dater is just to minimally error correct chronograms to make them compatible with all the given relative time constraints, right? So that's really the key idea behind Dater. And by minimal error correction, we mean that we want to adjust the original branch lengths or node ages as little as possible to satisfy all the constraints. So Dater basically takes as input a bunch of chronograms, right? The posterior samples. So for example, maybe 1,000 chronograms or so, which are constructed just based on absolute time calibrations, or node age calibrations. And it takes as input a set of relative time constraints. And then it uses a constraint optimization framework to minimally error correct these each of these input chronograms, each of the M input chronograms to satisfy all the given relative constraints. Right. And then once we have these error corrected chronograms, we aggregate across all of them to get one representative sort of median error corrected chronogram. And the output is, again, just the final aggregated chronogram as well as the individual error corrected chronograms. So that's basically what data does. Obviously, there is no obvious, clear or standard way to define what minimal error correction means, right? What we mean by minimal change. So this can be defined in many different ways. So for example, we might want to minimize the squares of the differences in branch lengths between the input and output chronograms. You might want to, instead of looking at absolute differences, maybe you want to look at the ratios of the branch lengths, right? The change in the ratio of the ratio of the branch length. That's one way to do it. Or we might focus on the actual node dates themselves. You might want to minimize the change in node dates, right? So there are many different ways to look at this. And data implements 3 objective functions that capture exactly the 3 kinds of intuitions I talked about. So one of these objective functions is the minimum squared branch length deviation. We refer to it as SBD. Another is to minimize the squared log ratio of branch length, that's SLRB. The other is to minimize squared date deviation, right? And again, all of these are sort of squared simply to penalize bigger changes more than smaller ones. So this is the objective function for SBD. Again, you can see this is like the difference of branch lengths summed up over all branches. Take the square, and there's some sort of a weighting factor here to penalize big changes on shorter branches more. Right. So if this is the input chronogram, it violates this constraint. Then in the output by applying this objective function might look like something like this. Right, and so on. We have the other objective function as well. So SLRB looks like this. It might produce a slightly different final dating. SDD looks like this. It might again produce a slightly different final dating. So we applied this to a bunch of simulated data sets, and more interestingly, to a real biological data set of 170 cyanobacterial genomes. This is the data set from the rejection sampling approach from Fournier et al. 21. And so here we had a high confidence species tree or a species phylogeny of 170 leaves, and we had the analysis under 6 different molecular clock models, right? So uncorrelated gamma with and without birth date priors, autocorrelated models with log norm autocorrelated model with and without birth date prior, and the CIR model again with and without birth date priors. So 6 models, 1,000 chronograms for each, and 24 manually curated HGTs or relative time constraints. And this was a very deep tree, like the root is almost 4 billion years old. So let me quickly rush through my slides. So the first thing we noticed is that the sample chronograms, if we just sample them without error correction, they all violate lots of the relative constraints, right? So even the best model here, CIR, nobody violated like 3.5 constraints on average. And the worst ones violated much more. So there's a lot of incompatibility going on. And in fact, the magnitude of this incompatibility is pretty large, right? So for the violated date constraints, the violation is on the order of hundreds of millions of years, right? It's a pretty big violation. And so one thing we noticed was that data can achieve compatibility using really small adjustments, right? So for such a deep phylogeny, data can achieve full compatibility with all the relative constraints by making very small adjustments to the node dates and to the branch lengths, right? And even if you look at the percentage change in branch lengths, again, that's very, very low, like about 1.69% for this model. In the worst case, like 7% for the worst model. So it's very effective. It just slightly changes the tree to satisfy all the relative constraints. We also found that maybe SLRB and SBD are preferable to SDD. I'll sort of skip over this result, but that's what we found. SDD can really change branch lengths quite drastically.
All right, Dr. Bansal, I'm going to have to stop you there. Sure. That is the end of your talk slot. Now, we do have a withdrawn talk in this period for the next 15 minutes. So, you can stay unmuted if people want to ask questions to either of our any of our 3 speakers who have already gone today. feel free to put up a hand during this time, and we can accept questions. And we can also have people put questions in the chat. Okay, so it looks like Matthew has a question, so let me ask you to unmute.
Hi, thank you for the talk. This question is for Dr. Bonsall. I'm wondering if it would be a relevant consideration to take into account confidence in the relative constraints. I could imagine there are scenarios where you're highly confident in the evidence for a particular gene transfer event and less confident in, a different one.
Indeed, that's a great question. So first of all, I apologize for not being able to finish my talk, but I think you heard most of it. So that's a great question. So yes, we don't do it right now. Right now, we insist on satisfying all the constraints. But in future work, we do want to have some sort of a model that can maybe weigh how much you are changing with respect, maybe dropping a few of the AGT constraints can get you a really good chronogram without too many changes, right? So yes, it would be some sort of a soft bounds approach, right? The soft bounds approach that's there with typical chronogram inference methods, right? We want to add that sort of thing for this as well, yeah.
Great, thank you.
All right, so if anybody had just joined us, I noticed we got a couple more participants as we were taking a question. We do have a withdrawn talk in this slot. And so if anyone wants to ask questions for our three speakers who have already spoken, you are certainly welcome to do that. I'll be here to moderate and to unmute people as questions arise. If you're feeling shy, you are certainly welcome to ask questions in our chat window. I'm going to go ahead and pause recording for the next little bit here. And if anyone needs a break, we are going to resume at the top of the hour with Dr. Liu's talk.
Awesome.
There we go.
All right. Well, thanks everyone for attending my talk. Also, thanks to the organizers of this fantastic conference. With apologies to Ralph Waldo Emerson. Yeah, that's this long title. Let me just summarize it as really my talk is about a new method for estimating phylogenetic support, which I'm going to try to convince you basically is an improvement upon the state of the art. Before I begin, I'd just like to acknowledge the co-authors on this work. This includes multiple current and former students in my lab. So I guess I'd like to begin talking just about phylogenetic support itself. It's really almost a de facto standard anytime you try to reconstruct and talk about a phylogeny in any scientific study. And the idea behind it is really you want to assess reproducibility, often using some classical statistical burst sampling methods, and in essence trying to effectively just place confidence intervals on a phylogenetic reconstruction. And by far the most popular, most widely used standard still remains the phylogenetic bootstrap method as introduced by Joseph Felsenstein in 1985. Very influential work, which according to a 2014 survey is among the 41st most cited papers in all of history. In brief, what he proposed was to do the following. You take an input, multiple sequence alignment, and a phylogeny that you want to annotate with phylogenetic support. And what you do is you take your input MSA and basically just get more resampled data sets from it using the standard bootstrap method proposed by Efron, which basically says the following. You take your input sequence of observations here, all the sites in your alignment, and then you sample from your data uniformly at random with replacement. And there's a key assumption here. Basically, you're assuming that your input observations here, sites in the MSA, they must be independent and identically distributed, or IID. And once you have your resampled data sets, you basically perform re-estimation on each of them, and you compare your re-estimates against your original estimation to calculate reproducibility. And it's quantified as follows. For each bipartition that appears, or edge that appears in your input tree, or phylogeny, you just calculate the fraction of re-estimated phylogenies that also display that bipartitioner edge. Now, as I mentioned, it's a critical assumption to assume that your data are identically distributed and independent. And in fact, there's a bunch of fundamental reasons why this is not the case for biomolecular sequence data. Among them are things like biomolecular structure and function, genetic recombination, and also evolutionary processes such as sequence insertion and deletion events, which basically cause this assumption about IAD data to be violated. And there's some real important consequences to this. And fundamentally, what this means is that Felsenstein's application of bootstrap sampling to biomolecular sequence data, it has some major consequences. And then the question is, well, can we do better? And one of the earliest attempts to try to do better, to try to come up with alternative techniques, not for that problem, not for assessing statistical confidence during phylogenetic reconstruction, but actually for assessing reliability of multiple sequence alignment, really goes back to the early 2000s to Landon and Grauer, who basically proposed the following kind of clever trick, which is referred to as the heads or tails technique. Basically, what they said is as follows. You take kind of the alignment problem, which looks as follows. Take your inputs, the unaligned sequences, run it through your alignment method. It gives you an alignment as output. What Landon and Grauer says, well, a nice reliability check is you should, in principle, get the same result if you basically read your input sequences from right to left rather than from left to right, as shown here. It should be the same, but actually, often it wasn't based on their assessments. And so this is indeed a pretty nice, kind of baseline sanity check, if you will, for alignment estimation, this heads or tails technique. But one weakness that you can see here is that you really only have two things to compare together, two replicates in effect. And so what we wanted to do was to think about if we could come up with a better resampling technique, which gave you more replication beyond just these two. And so to kind of make progress on this question, we actually made use of several different insights. The first was, look, if heads or tails said that, it shouldn't make a difference whether you read from right to left or left to right, what we realized is that same idea could be applied at the interior of the sequences in addition to the end. But to do so in brief, I'm going to show you kind of these ideas using a guide alignment. It's only really used for kind of synchronizing things during the subsequent algorithm that I'll propose, but otherwise unneeded. Anyways, within this aligned sequence data, the way you apply this first insight is, well, let's just read from right to left or left to right at the interior of your aligned sequences rather than from the starter ends, okay? And so if I did from this particular point, reading right to left, I basically get this unaligned sequence data running the reads in that order. But then if you're comfortable with that, when you hit the ends of the sequences, basically we can just reverse our kind of reads of the data at the starts or ends. And this is basically equivalent to reflection at the start or ends of the sequences. The alignment problem that we care about and that Landon and Grauer cared about, it'll still be meaningful for globally alignable sequence inputs if you do this reflection. This is the second insight that we made use of. And finally, if you're comfortable with all that, not only can we reverse the redirection at the ends of the sequences, but we in fact can do that at the interiors as well for randomly chosen reversal breakpoints, which are chosen with some probability, which we'll parameterize just in a few seconds. And what you get as a result is a procedure which basically takes the form of a random walk conducted directly on your sequence data. We refer to this procedure as RAR, which is short for random walk resampling. And this is basically a new sequence-aware non-parametric resampling method. It was published just recently in Oxford Bioinformatics. And in the end, it basically generalizes bootstrap resampling and the mirrored inputs idea that Landon and Grauer proposed in their heads or tails technique. And this is the fundamental kind of new contribution that we utilize to get better phylogenetic support estimates. Our RAR resampling technique is parameterized by a few things, among them the reversal probability gamma, which I just talked about a second ago, which controls whether or not you reverse the redirection at any point during a random walk. And finally, the termination criterion for the resampling itself. Now, in our first study, that 2021 study, we actually applied this new resampling technique to phylogenetic support estimation in the form of the following pipeline. From an input of unaligned sequence data, you basically get your annotation MSA and tree. This annotation MSA is used to run the RAR random walk process as I just described it. You get a RAR replicate set of unaligned sequence data. That's critically important, in which the most important kind of property of the RAR walk is that you preserve neighboring nucleotides or bases as you do the random walk. And so there's this critical aspect of the preservation. Any pair of nucleotides or bases or letters that appear in your input data will also be guaranteed to appear sequentially in any RAR resample replicate. Once you have the unaligned RAR resampled data, you realign it, and then you re-estimate the tree on the re-estimated alignment. And of course, as with any resampling analysis, you do this a bunch to get a bunch of re-estimated alignments and re-estimated trees. And then using the re-estimated trees, we compare this against the original estimate tree and I guess alignment to basically calculate phylogenetic support in the traditional way. You simply calculate the support of each branch based on the proportion of re-estimated trees that also display that branch. And this is kind of our first application of RAR to phylogenetic support estimation. We then conducted a comprehensive benchmarking, a performance benchmarking study, which utilized both simulated data sets with a varying range of data set sizes and evolutionary divergence, as well as some very nice empirical benchmarking data courtesy of the Comparative RNA website. We compared against state-of-the-art methods, including Bootstrap and a newer method referred to as the transfer bootstrap expectation method, published by Lemoyne et al. In 2018. We assessed all of the methods based on type 1 and type 2 error, runtime, and main memory usage. I'm only going to quickly summarize the findings from the simulation study. We basically found that RAR either returned comparable or often better type 1 and type 2 error compared to these state-of-the-art methods. And its advantage tended to grow as data sets became larger and sequence divergence increased, and correspondingly, tree estimation became more difficult and more challenging. But this came at a trade-off, I must be clear about. So the advantages in terms of type 1 and type 2 error, they did actually also come with some scalability costs. So there was, although there was comparable memory usage across the different methods, RAR did require more runtime overhead than the other methods, in part due to the need to perform re-estimation of alignments. And in the remainder of the few minutes that I have, I just want to talk briefly about the empirical study that we also conducted using RAR support estimation. We actually went back and looked at a pretty celebrated study from Lamachani et al. In 2015, and also one of their follow-up papers, which looks at a very famous model of adaptive evolution and actually other fundamental evolutionary processes. I'm referring to Darwin's finches, which Darwin studied. And so we used RAR to basically look at or reanalyze a whole genome sequence data set from this 2015 study of Darwin's finches. Here's just a map of the Galapagos Islands from where the sampled data was drawn from, came from a bunch of different species, from a bunch of islands within the Galapagos Islands itself. And what we did was we basically began by re-estimating a species tree using their species tree estimation approach. We actually got a somewhat different result. Here I'm showing you that annotation tree that we reconstructed. The names of the taxa aren't particularly important, or the species rather. I'm actually coloring them by different groups, including tree finches, sharp-beaked ground finches, etc. And one clade in particular here highlighted in dashed green, I'm highlighting the tree finches clade because this is the main one in which we observe differences based off of our reconstructed species tree compared to the 2015 paper that Lamachani et al. Published in, I believe it was Nature or Science. So there is a difference, a topological difference between our reconstructed tree and theirs here within this tree finches clade. In addition to this topology, I am showing you also phylogenetic support values calculated using the traditional phylogenetic bootstrap method as proposed by Felsenstein. Basically, you can see it's just very highly supported almost everywhere. You see 100% support. In contrast, when you run RAR, what you see actually is fairly similar support values calculated using the approach that I just described, except here within the tree finches clade. There are some strong differences here. Basically, these two branches, these are some of the branches that our species tree estimate differed from the 2015 publications reconstruction. And so, in part, we would claim that we interpret this to be that RAR essentially is attributing some of the phylogenetic reconstruction uncertainty due to these issues of essentially alignability within this particular clade. And so in summary, what I have talked about briefly is our new risk sampling method referred to as RAR, which takes the form of a random walk to basically do sequence-aware statistical risk sampling of biomolecular sequence data. We studied the performance of RAR using simulated empirical benchmarking data sets and showed that it typically outperformed the state-of-the-art in terms of type 1 and type 2 error. Did come at a cost, though, in terms of runtime. And then actually, our empirical studies seem to suggest that sequence insertion and deletion processes may have played some kind of important role in terms of phylogenetic estimation uncertainty within this very celebrated system of Darwin's finches. Looking forward, I'll briefly mention just some ongoing work. We are also studying other applications of the RAR sampling technique, also thinking more about this trade-off between kind of accuracy and runtime overhead, using kind of traditional computational techniques to help ameliorate this trade-off, things like parallelization. And finally, we're actually also thinking about applications of our technique to things outside of computational biology and bioinformatics. I'd like to acknowledge the funding agencies that supported this work. And I guess that brings me to the end of my talk. If you're interested in data, software, I'll point you to this, this open GitLab repository. We can find all of that. You can also find my contact info on my website. Feel free to reach out to me if you have any questions. Thanks very much.
Excellent. We do have time for a question. So if anyone would like to ask anything, you're certainly welcome to raise your hand and I will request that you unmute. And Matthew, if you want, you can get your slide share up while we take questions. All right, well, you can go ahead. Our final session or our final talk of the session is Matthew Moreno, and that will, at the end of his talk, will mark the end of this session. All right, take it away.
Great, thank you. Hi, everyone. My name's Matthew. I'm a new postdoctoral scholar at University of Michigan, doing computational work with Luis Simon. And today I'm going to be talking to you about work I did collaborating with Emily Dolson at Michigan State University and one of our undergraduate researchers, Santiago Rodriguez Papa. I'm really excited to talk to you about recent work laying the foundations to use phylogenetic methods as a tool to increase the observability of really large-scale many processor evolution simulation experiments. So this work is in the field of digital evolution, which refers to the use of agent-based computer models to simulate the evolutionary process. This ranges everywhere from evolving bit strings on an NK fitness landscape to evolving full-fledged genetic programs, self-replicating genetic programs like you might be familiar with the Avita system. And the fundamental idea is to instantiate the evolutionary process in a computer program to enable experimental manipulations and experiments that may not be possible otherwise due to the unique capabilities of the simulation in having fast generational turnover, complete observability, and kind of the ability to perform arbitrary experimental manipulations. As you might expect, there's a really rich history of application of phylogenetic methods in the field of digital evolution. Of course, phylogenetic analyses can give you kind of a play-by-play account of how different traits in a population arose, but they can also in principle be used to detect more general evolutionary dynamics like the presence of ecological dynamics, to characterize selection pressure, etc. As computing hardware progresses at a rapid rate, there's beginning to be the possibility of really scaling up the simulations that we're able to perform to do these experiments. And this raises kind of an issue, because as these experiments get to larger and larger scales, it becomes intractable to completely observe the system. And so we need to develop methods to be able to analyze these really large scale simulations without having to observe the system in its entirety. And phylogenetic methods are a promising step in this direction. However, there's a fundamental issue with applying phylogenetic methods to large-scale evolutionary simulations, which is that the existing methodology in digital evolution for phylogenetics work relies on a perfect tracking process. In this type of process, all of the reproduction events in simulation are logged, and then after the fact, you can go through and kind of stitch together all of the parent-offspring relationships and perfectly reconstruct the phylogeny. However, as you scale up to many processor experiments, it becomes kind of intractable to continue this perfect tracking approach. You can kind of think of this like a group of people trying to all share a filing cabinet, where you have a small group of people. This is reasonable. You're not going to have to wait in line too long to use the filing cabinet. But if you have a big crowd of people that are all trying to use the same filing cabinet, it quickly becomes unworkable. And then when you introduce the possibility of lost or of lost data, which is a frequent occurrence in really large scale simulations, this perfect tracking approach becomes even more difficult. And so our approach to getting around this issue is inspired by the really spectacular success that evolutionary biologists working with natural systems have had in doing phylogenetic analyses in systems where there isn't perfect tracking, i.e. Using a reconstruction-based approach where you just gather the set of genomes from extant organisms at the end and then do sophisticated analyses in order to infer a guess or a probable phylogenetic history of the sampled organisms. And so this current project builds on recent work, which kind of tackled a kind of fun puzzle of in digital systems, we have the ability to design how things work. And so an interesting question that we had to tackle is, how should we design digital genomes in order to maximize the accuracy and efficiency of phylogenetic inference? And this is kind of like a interesting backwards problem where we get to actually design the data that we want to perform inference on, and not just the algorithms. So here's a link to kind of a little blurb on that if you're curious about that, but I'll kind of gloss over that for now. But this current work is looking at actually now that once we have these phylogenetic reconstructions, how can we use them to infer evolutionary dynamics of these systems? And so we're going to talk about looking at signatures of ecology, selection pressure, and spatial structure in phylogenetic metrics. And there's other work that we did as part of this project that not talking about today, which is looking at how reconstruction error biases phylogenetic metrics and seeing kind of what levels of reconstruction accuracy are necessary to drive that reconstruction bias down to near zero for different metrics. We used 4 phylogenetic metrics, trying to kind of cover the scope of different aspects of phylogenetic structure, looking at internal node count, coalesce index, pairwise distance, and evolutionary distinctiveness. And in order to perform our experiments, we used a really simple model system because we needed to be able to compare perfectly tracked phylogenies with reconstructed phylogenies. And so we just used agents that had a single genome value, which just directly represented their fitness. So it was a continuous floating point value, and agents that had higher genome values would have a competitive advantage against agents that had lower ones. And we applied Gaussian mutation every generation to our population. We were able to simulate 250,000 generations of evolution with a population size of about 30,000. And then we had 3 different levers that we used to manipulate the evolutionary dynamics. We manipulated selection pressure by changing the tournament size of the selection process in the algorithm. This basically affects how fast fit solutions are able, or fit agents are able to take over a population. We also used a simple island model with a fixed migration rate to simulate spatial structure, and a simple niche model with a where organisms would be divided into either 4 or 8 niches, then only compete within their own niche and have a small probability of being transferred and invading a different niche at a rate that where we would expect 1 invasion per about 1,000 generations. And so here is a summary of the phylogenetic outcomes measured by our 4 phylogenetic metrics on the y-axis for our different treatments. So the different treatments are on the x-axis, with the kind of control treatment being on the left, and then relative change in the phylometrics being shown with the color coding. So red indicating an increase in that phylogenetic metric, and blue indicating a decrease in that phylogenetic metric, and then the stars indicating statistical significance with 40 replicates. So kind of some interesting things to highlight here, which is that there appears to be kind of interesting nonlinear relationships with some of these, where, for example, when we weakened selection and when we strengthened selection, we saw decreases in the coalesce index and the mean pairwise distance. But what really caught our attention was the really strong effect of spatial structure on these phylogenetic metrics, and the relatively weaker effects of ecology, where we're only able to detect significant changes in 2 of our metrics for our ecological treatments. And so here's some example phylogenies on the bottom left, phylogeny from one of our ecological treatments on the bottom right, one with the spatial structure with that island model. And you can see the spatial structure is really increasing the richness of the phylogeny. And so we became interested in whether you could, because of the strong effect of the spatial structure, whether you would still be able to detect ecology in the presence of spatial structure. So we combined the ecological conditions and the spatial structure conditions, and then performed comparisons of with ecology versus without ecology. And we actually surprisingly found that the presence of the spatial structure enhanced the detectability of the ecology. So on the left here, I have the distribution of the coalesce index in the absence of spatial structure, and then on the right, in the presence of spatial structure. And I've highlighted in yellow the control treatment, and then the bottom 3 bars are the different ecological treatments. And you can see that there's a much more distinct separation between the ecological treatments and the control in the presence of spatial structure. And we saw this pattern across all 4 of the phylogenetic metrics, in particular in the 2 metrics where we weren't able to detect any ecological effects without spatial structure. In the presence of spatial structure, we were able to detect ecological effects in these phylogenetic metrics. So in the interest of time, I'll kind of fast forward through these and wrap up here. To summarize what we did, we looked at evolutionary dynamics of ecology, selection pressure, and spatial structure, and evaluated their effects on different phylogenetic metrics. And we saw kind of some nonlinear complexity occurring in these relationships. And we found that spatial structure had a really strong effect on these phylogenetic metrics, but it actually enhanced the detectability of the relatively weaker ecology on these phylogenetic metrics. So I'll go ahead and wrap up and acknowledge my collaborators and my advisors, and I'm happy to take questions. Thank you.
Excellent. So it looks like we already have a question. Let me invite Michael to unmute himself to ask that.
Hello, I wanted to know if you would see the same niche effect size increase if that you saw with the spatial segregation, if you reduced the frequency of niche invasion.
Yeah, so we did have a treatment. So we had a 4 niche ecology and an 8 niche ecology. And then we had weak 4 niche ecology, which did the opposite of what you're suggesting, that increased the probability of niche invasion. So under this weak 4 ecology treatment, niche invasion was 1,000 times more likely. And we weren't, it looks like at least in this particular case with the ancestor count, we weren't seeing any differences in the ancestor count with that difference in the niche invasion probability. And I don't think we have time to kind of go through the other ones, but that would definitely be interesting to look at. Yeah, thank you.
Thank you.